things are more devastating than diseases that leave the mind intact. My story begins not with science, but with love. In 2015, my grandmother, my Nannan, was diagnosed with amyotrophic lateral sclerosis, or ALS, more commonly known as Lou Gehrig's disease. Now, if you've never heard of Lou Gehrig, he was a baseball player, and his diagnosis brought national attention to this disease. ALS doesn't just take life. It takes it piece by piece. In less than 2 years, it snatched my Nannan from me. But what if a bacterium and a virus hold the key to curing ALS? By her final year, my Nannan required a non-invasive ventilator just to breathe. Yet ALS could never take away her smile or diminish her spirit. Even during those final few months, her impact was undeniable. Her students, family, and friends came from all around the world to visit her one last time, filled with stories, gratitude, and love. She taught me that strength was often quiet, steady persistence. My Nannan was a mother, a grandmother, a teacher, a fighter, and a source of light. While ALS claimed her body, it never stole her identity or the legacy she left behind in the hearts of those who knew her. For me, this disease is personal. It has a face and a name, my Nannan. Knowing someone with ALS reveals a difficult truth. Compassion on its own is not enough. Effective tools make the difference, and it's here where the science comes in. So you might be wondering, what exactly is ALS? ALS is a neurodegenerative disease, and despite ongoing research, a treatment remains elusive. ALS is often more than just a diagnosis. It becomes an imprisonment of the body. ALS targets motor neurons, the cells responsible for movement, speed, swallowing, and breathing. But over time, as these cells fail, ALS progressively takes over these functions. All this while your mind is awake and your thoughts are clear. Imagine the plight. But ALS doesn't just affect the person diagnosed. It quietly reshapes the lives of their caregivers too. Family members who become nurses, translators, and constant companions. They watch someone they love slowly lose the ability to move or to speak, while doing whatever they can to still hold on to that person. Now we ask, why don't we have a cure for ALS? But what we don't often understand is that ALS doesn't have just one single cause. It often results from single genetic mutations, and those differ between patients. Around 10% of the ALS cases are familial, meaning that they result from genetic mutations. The other 90% appear sporadically without any clear family history. In those sporadic cases, scientists have still been able to identify only a few of the causes for ALS. The rest remains a mystery. But instead of being overwhelmed by what we don't yet know, let's first start with what we do know, the familial or the genetic ALS. Two patients may look the same, but their DNA tells a completely different story. A disease with so many different origins is difficult to treat with just one single solution. Scientists have searched the human body for years, but recently, a breakthrough happened from an unexpected source. Long before humans, bacteria were already fighting wars against bacteriophages, the viruses that attack them. You can imagine a bacterium like a tiny house, and a virus like a burglar trying to break in and hijack the house. But instead of forgetting the attack, the bacterium keeps a small piece of the virus's DNA, like a genetic fingerprint, stored in its own DNA. When the same virus returns again, the bacterium will be able to recognize it and fight it more effectively. We call this memory system CRISPR. Then, a pair of molecular scissors, Cas9, comes and cuts the DNA at an exact spot. Soon, scientists realized that they could reprogram the system. Instead of targeting viral DNA, we could target a specific genetic mutation. In some forms of ALS, in which the cause is just from one single genetic mutation, the CRISPR-Cas9 system offers a remarkable possibility: editing the mistake itself. What I'm about to share with you is still a work in progress. There's still no cure for ALS today, but for the first time in a long time, there's real light at the end of the tunnel. And it may not be as far away as we once thought. Looking at another analogy, let's depict your DNA as a massive library. Each chromosome is a book. Each gene, like the SOD1 gene, is a chapter in that book. And DNA is just a long string of letters. Now imagine the CRISPR-Cas9 system trying to target the DNA at an exact spot. The system uses a tiny bookmark or road sign called a guide RNA to help it find that spot. The bookmark is just 20 letters long. Just 20 in a book with billions of letters. And those 20 letters have to match the DNA almost perfectly, especially the first 10 letters near the cutting site. If even one important letter is misplaced, the scissors will not cut. Therefore, when we design a guide RNA, we're essentially writing a 20-letter bookmark that has to match one place in a 3 billion letter book and nowhere else. And that's why precision matters. However, not all of the sentences actually make it into the final printed copy, and the ones that do are called exons. They're what tell the cell exactly how to build the protein. The rest are called introns, and they're left out before the final message is produced. If a typo occurs in an exon, it changes the meaning of the entire sentence. And it's here where disease can begin. But the challenge is that in diseases such as SOD1-linked ALS, that typo differs between patients. A guide RNA designed for one mutation is not necessarily going to work for another one. For me, this disease becomes personal because of my grandmother. When I learned that certain mutations in the SOD1 gene, such as the D91A variant, are linked to inherited ALS, I started reading whatever I could. Researchers found that around 12 to 20% of familial ALS cases are caused from the SOD1 gene. And mutations such as the D91A mutation are linked to Asian populations. And then I couldn't stop thinking, what if we could edit that one mistake? I cannot yet edit genes in a lab, but I can model the battlefield. My research uses computational tools to design and rank CRISPR guide RNAs. I'm essentially mapping the coordinates before the Cas9 scissors can even touch the DNA, because before we edit the sentence, we have to know which word we're going to cut. But how does the guide RNA even enter the human body? A virus, the very thing we were taught to fear, becomes part of the solution. Scientists use a weakened modified virus called adeno-associated virus, or AAV. All of its harmful parts are removed, and instead, it's loaded with new instructions, the CRISPR guide RNA. Instead of hijacking the cell, this virus simply drops off the package. It delivers the editing tools to the exact place in the DNA that needs correction. The invader becomes the courier. So, let me ask you one final time. What if a bacterium and a virus hold the key to curing ALS, not because nature's against us, but because nature's been preparing us? The smallest organisms on Earth have taught us how to remember, how to recognize, and how to repair. From their ancient battle, we gained a tool powerful enough to rewrite disease. My grandmother lost her battle with ALS, but her story is not finished. Because when we design a guide RNA, when we map a mutation, when we move one step closer to precision, we're refusing to accept that this is the end of the story. And as we honor the patient, we also honor their caregivers, the family members who give their strength, their time, and their hearts day after day, even when hope feels distant. Maybe the cure for ALS will not come from something grand or dramatic. Maybe it will just come from something microscopic.